Hello, everyone. Thank you all so much for joining today. Just wanted to let you all know that we're going to start promptly at 12 p.m. Eastern Time. Hello, everyone. Thanks so much for joining. We're going to begin promptly at 12 p.m. Eastern Time, so in a few minutes. All right, we're going to go ahead and get started. So hello, everybody. Thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency, collaboration, and highlight the incredible omics research underway both within and outside of NOAA. The seminars from this series are all made available on YouTube and posted up on our omics website. You should all be able to access that website. There's a link to it in the chat right now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end of today's presentation, I will read the questions out loud for our presenter to answer. One last point here, the NOAA Omics Seminar Series also takes place on the third Wednesday of each month at 12 p.m. Eastern Time. However, seminars like these ones sometimes may be scheduled outside of this time frame, so please check the website to stay up to date on when our omics presentations are taking place. All right, so with that, our presentation today is titled Using Environmental DNA to Examine Artificial Reef Fish Assemblages. If you build it, will they come? Presented by Dr. Kyle Pillar. So Kyle, I'm going to give you screen sharing permission now and let you get that set up while I introduce you to our audience. Okay, so Dr. Kyle Pillar received a BS degree from the University of Illinois, an MS degree from Southern Illinois University, and a PhD from Tulane University. He completed a 2-year postdoc at the University of Wisconsin Center for Limnology and Wisconsin DNR. He recently completed his 19th year as a faculty member at southeastern Louisiana University. He has an active research program in which he uses a variety of molecular approaches to address questions in fish diversity in the southeastern United States, Mexico, and Central America. He has published 61 papers on molecular systematics, population genetics, environmental DNA, and morphological variation of marine, diadromous, and freshwater fishes. He has trained 23 MS students and 2 postdoctoral researchers since 2003. So for more information, you can check out his website at kylepillar.com. So Kyle, thank you so much for presenting today. Just wanted to let you know that we can see your presentation. It looks good. And I will now pass it off to you.
Okay, thank you to Catherine and the Omics group for the invitation to come speak about 2 projects that we've been working on in my lab the last couple of years. So before I get started, I need to acknowledge that these 2 projects were conducted by 2 different master students in my lab, Erin Crollo and Megan Ryba. So they deserve a lot of the credit for the things I'm going to show you. They collected the data, they analyzed the data, and I'm just here, I'm just a presenter. So without further ado, let's go ahead and get started. Okay, so today I'm going to talk about artificial reefs. I assume most of you are familiar with sort of what their functions are, but they're essentially designed to mimic natural reef habitats. They're used as a mitigation tool worldwide, but there's been a lot of effort in the US over the last 10 years to sort of construct and build artificial reefs all over coastal regions and somewhat in freshwater as well. They're primarily deployed to increase fisheries, either to create new habitat, replace lost habitat. Usually the replacing of lost habitat is through some anthropogenic modification that has occurred in various regions, or also to enhance existing habitat. So when you think about what is an artificial reef, it's kind of a broad definition. Seaman in 2000 said, an artificial reef essentially is one or more objects of human origin deployed purposely on the seafloor to influence physical, biological, or socioeconomic processes related to living marine resources. This has been expanded to obviously freshwater, as I mentioned, but also to brackish and estuarine habitats. The interesting thing is that what we have used as artificial reef habitats has changed over the years. Initially, there was sort of the idea that any type of material, hard material would work, and we call these materials of opportunity. Later on, as sort of we learned more about the different types of artificial reefs, there would be more select materials of opportunity, followed by prefabricated or engineered materials, and to now what we call designed materials. So there's this interesting evolution of materials and types of reefs that have been deployed over the last 10 or 20 years. I'm just going to briefly touch on a few of these that we sort of have in my region in coastal Louisiana and the Gulf of Mexico. Materials opportunities, again, these are the earliest reefs. Simply, these are any available material, hard material that could be deployed into the environment. A lot of people view these materials of opportunity reefs as sort of the out of sight, out of mind types of reefs. For example, a couple of the reefs in coastal Louisiana are essentially high schools, for example, and the bricks and concrete that were destroyed as a result of Hurricane Katrina. So rather than deposit those materials into a landfill, they basically took those and deposited them on the sea floor near the mouth of the Mississippi River as an artificial reef structure. Early on, there was concern that some of these materials could be toxic. Obviously, from the images here of these boats or this tank, obviously the goal would be to remove or eviscerate any of the potentially toxic materials within these structures, but that was not always the case. Concrete materials are a favorite, at least in my part of the world. Obviously, concrete is cheap and readily available, not just here, but globally. It's similar in composition to natural coral or limestone. We can mold it into any shape. It is also a material of opportunity. Things like bridges, sidewalks, old culverts. We see a variety of different types of concrete-based artificial reefs that have been deployed, at least in the Gulf of Mexico, over the past 10 or 20 years. What seems to be the preference nowadays are prefabricated or engineered artificial reefs. Again, here in my region, reef balls seem to be the most popular one. That's the picture on the bottom there. They're comprised of non-toxic materials, or constructed of non-toxic materials, and they're often deployed in clusters with sort of open holes that allow invertebrates to colonize the inside and the outside and offer sort of refugia for small non-game fishes. So these seem to be sort of the artificial reef of preference, at least in the Gulf of Mexico right now. And limestone is another one. This is really cheap to deploy limestone reefs. They're relatively stable, although over time they can fill in with sediments. But early on, they offer a really high degree of habitat heterogeneity, both for the colonizing invertebrates as well as small non-game fishes like gobies and blennies, things like that. As I mentioned, they're cheap and they're relatively easy to deploy. So this is a popular one in my region as well. And finally, oil and gas structures and materials. This typically consists of decommissioned oil and gas rigs that are often donated by petroleum companies, used typically as part of a tax relief. When these structures are upright and in use, at least in the Gulf of Mexico, recreational fishermen take advantage of these areas because they are really good at attracting fishes in an otherwise sort of open aquascape. Fishermen go to these, they flock to these for catching things like red snapper and grouper. And so the idea was if they're no longer in use, let's just leave them there. And that's sort of been a big discussion. Once an oil rig is no longer in use, should we leave it there for something like this? Or is it better to remove it for safety reasons? That's sort of a big debate. And sort of what came out of this was this Rigs to Reefs program, where they are deploying these types of materials all across the Gulf of Mexico. So in Louisiana, or at least the coastal Louisiana, the artificial reef program was initiated in 1986. This is a sort of an ARC shapefile that was provided to me by Wildlife and Fisheries that shows the position of artificial reefs in coastal Louisiana. We have inshore, nearshore, offshore, and deep water reefs. Most of what I'm going to talk about today are the inshore artificial reefs. So coastal Louisiana is the second largest seafood supplier in the nation. There are roughly over 4 million recreational fishing trips annually. So there's a lot of money coming into coastal Louisiana for recreational fishing. And so as a result, the state wanted to augment that. So they deployed artificial reefs, not only to increase fishing opportunities for the local citizens, but also to restore lost habitat and improve fisheries in certain parts of coastal Louisiana. Initially, the artificial reef program in Louisiana was kind of this, I call it the field of dreams approach. If you build it, will they come? And sort of that's sort of the approach that was originally applied. They would deploy these reefs with really not an easy way to assess, are they being used by fishes? What species are using them? Are certain materials better than others? So that's sort of where I came onto the scene. So in coastal Louisiana, for the inshore artificial reefs, there are 33 inshore reefs along coastal Louisiana. Most of these are limestone reefs or crushed concrete. And then in the last, again, 10 or so years, reef balls have come on the scene. These are typically deployed in shallow water environments, typically less than 5 meters in depth for the most part. Again, these are frequently used by Louisiana fishermen because the state does advertise the latitude and longitude coordinates for all these reefs. So the fishermen know where they are and they go and fish at these structures. So one of the challenges, as you might expect, of artificial reef sampling is how do you assess fish assemblages? How do you monitor fish assemblages or the success of a particular type of a reef? Well, clearly there's a variety of approaches that have been deployed. We can do visual surveys, gill net and trawl net surveys, substrate trays as just some examples, but these are not necessarily the most ideal ways to assess fish assemblages at artificial reefs. Gill and trawl nets, obviously you have to consider or deal with potentially of entangling your nets or destroying the reef as you're dragging a trawl around it or near it. So that's problematic, it's not ideal. Visual surveys, again, in this region, turbidity is really high. So sometimes it's really hard to see fish species accurately in artificial reef environments simply because of the high turbidity. Coupled with that is the identification expertise of the diver or the ROV and then the person assessing the fish community. Is visually, is it clear enough for someone to really detect what species there? Clearly, you're going to miss things, particularly non-game fishes. And then substrate trays, they focus on benthic species. So you're missing the pelagic species that would be on and around the artificial reef. So until recently, there's really no good way to assess artificial reef fishes. So this is where I came in. I wrote a proposal for wildlife and fisheries to sort of assess, use this new technique at the time, an environmental DNA, non-intrusive method, focuses on filtering water and capturing free DNA in the environment. It's useful for both common and uncommon species. It's non-destructive. And with the approaches that we have available now, we can get a whole community or a whole fish assemblage sort of from one set of water samples from a reef. So we've employed an eDNA metabarcoding approach where we can get a whole snapshot of the fish assemblage or fish community with one water sample. So the first part, what I'm going to talk about, focuses on the paper that we recently published in Transactions American Fisheries Society. This was conducted by my former student, Aaron Crollo. If anyone's interested, I can send a PDF, but it's available on Transactions American Fisheries Society website. So this focuses on reefs in the northern Gulf of Mexico. So the big question we always have is what ray-finned fish species are present at these artificial reef habitats? And sort of within that larger broad question, we had 3 specific hypotheses that we wanted to test. First, with the introduction of reef structures that congregate fish from the surrounding area. In particular, do reefs harbor higher fish species richness relative to open water paired controlled habitats? The second hypothesis focuses on reef material. Do we see different reef fish assemblages, different levels of species richness for reefs comprised or constructed of different types of materials? We compared concrete, limestone, shell, oil and gas reefs in this study. We expected or predicted limestone reefs have the highest species richness, simply because of the high degree of habitat heterogeneity in the way that these reefs are constructed. And finally, we had reefs of differing age. Like the oldest reef we had was constructed in 1999. So we wanted to determine, was there a age influence on reef fish assemblages? And we hypothesized that older reefs would have more species, simply because they've had more time to establish a stable fish assemblage. So here's a map of the 9 reefs that we sampled in coastal Louisiana. We had 3 reefs that were constructed of concrete materials. We had 4 reefs that were comprised of limestone riprap, 1 shell reef, and 1 oil and gas reef. Our sampling was done in 2018, so we had reefs that were constructed in 1999 all the way up through 2017. So we use the Van Doren sampler. Simply we drop it down with both ends open, apply a weight that captures water right above the reef habitat. We took 3 reef samples and 3 control samples from each sampling area. We took abiotic measurements at each reef and control site, and we had 2 sampling periods, one in what we call the spring from February to May in 2018, and then another sampling event with the same 9 reefs from November to December in 2018. So the big question sort of we've always had is, what do we use for open water controls? Well, there was a paper published in 2015, Neves dos Santos and Zalman, where they assessed artificial reefs versus open water habitats. How far away from a reef do you have to get before it no longer becomes reef-associated community? And essentially what they found was that between 50 and 300 meters from a reef structure, the reef became no longer a reef type of a community, became a non-reef assemblage of fishes. So we sort of expanded that and went out about 500 meters between our reef and control habitats. And we sampled that slack tide for each reef when possible. In addition, there was an eDNA paper study published in 2018 where they basically showed that nearshore eDNA habitats, kind of like where we were sampling, the DNA there tends to be endogenous to the site rather than changing significantly with each tidal cycle. So we felt that by getting 500 meters away and with this eDNA study, we had a good representation of a true open water control and our true artificial reef habitat. So the approaches that we use, again, we capture water samples, we extract the DNA, we use the Qiagen Power Water Kit, we use 12S miteDNA primers from the Me et al. Paper. Sequencing was performed on HiSeq 2000, followed by analysis in the MyFish pipeline. For our data analyses, we use the data as presence-absence data. So we use a Soren's resemblance matrix, presence-absence data, and the greater than equal to 97% similarity matrix. That's part of the results from the MyFish pipeline. And all of our data was analyzed in Primer 7. So in terms of the schematics, what we did, we went to each reef, we took 3 water samples, extracted the DNA, then from those 3 water samples, we amplified 3 separate replicates for each one. So we had 9 PCR replicates for the 3 water samples from each reef. We diluted those samples to conduct the second round PCR as part of the Me et al. Protocol, incorporated the barcodes. We excised the gels, bands, cleaned them up, pooled them, sequenced them, ran them through the pipeline, and again, we get a greater than or equal to 97% similarity matrix, as well as an 80 to 96% similarity matrix. We focused on the greater than 97 in this study. So what do we find? Well, if we go back to the original hypotheses, would the artificial reefs congregate fish? And so here's what we found. From the spring 2018 sample, we recovered 24 total species across all the reefs with a range of 7 to 18 species per reef. For the paired controls, we got 19 total species with between 6 and 15 species per reef. If we think about species richness, at 3 of the 8 sampling events, control sites, open water pair controls, had more species than the reef habitats. And when we analyzed this statistically, we found no significant difference between species richness values at reefs versus controlled during the spring 2018 sampling, which was kind of interesting. The most abundant species was Gulf Menhaden, Brevoortia, followed by a mullet, a freshwater mullet, and then red drum was the 3rd most abundant species. But Menhaden dominated our reeds. In the fall of 2018, similar results. In this case, though, we got more total species at the controls, 24, than we did at the reef habitats, which we got 22. In terms of the ranges, they were similar across all the sites. Again, controls, we had more species at the open water controls than we did at the reef sites for 5 out of the 8 sampling events. Still no significant difference between reefs and controls. Again, the same top three species based on reed number recovered during the fall events as well. The second question, again, focus on reef material. Do the different reef materials have different fish assemblages with more or fewer species at each of these different types of materials? So if we compare the 4 reef types among our samples, limestone had the most total species, and this is what we predicted, with 21 total species. Much lower for the oil and gas habitat and the shell reef, 8 and 9 species total. That was for spring, and more or less the same thing for the fall samples. In this case, concrete and limestone reefs had the most total species with 18, where oil and gas had a total of 8. In this case, the shell reef had 15 total species, so a fair number more than it did during the spring habitats, or spring sampling. And finally, what about reef age? Again, we anticipated older reefs would have more established communities than younger reefs, so higher species richness. And we didn't find that. Actually, the oldest reef, 1999, is an oil and gas reef near Grand Isle. It had the fewest species here on the left of each one of these graphs. That represents the oil and gas reef. We find the highest species richness on average in sort of moderately aged reefs between 2012 and 2014. I think that this is probably an artifact that we simply had more reefs in that age group than younger or older. So we can't definitively say it's an age thing. I simply think that because we had more reefs to compare, that's why we had more species at those sites. But older reefs didn't necessarily have more species, at least for this analysis. We also want to look at assemblage structure. So when we compare a variety of effects, season, reef material, we get season and reef material as the only 2 significant main effects in assemblage structure. Now, if we focus on season, we see, and look at it in NMDS, we see substantial differences in fish assemblages associated with seasonal variation. Strong fall and spring fish assemblages during our sampling from these different reef structures. So what species are most responsible for the assemblage structure that we see between fall and spring assemblages? So we ran a SIMPR analysis, which basically tells you which species are contributing to the assemblage structure. The top 3 here are Esox niger, which is primarily a freshwater species, long-eared sunfish, Lepomis megalotis, a freshwater species. Threadfin shad, it does occur in estuarine or low salinity environments, but for the most part, it's a freshwater species. And when you get further down here, you get your typical marine or estuarine species, gaff-topped catfish, southern flounder, jack, things of that nature. But the top 3 were primarily freshwater species. And so that seemed to be driving some of the separation that we see in spring and fall fish assemblages at these reefs. So there seems to be a freshwater influence. And so if we look at the position of some of these reefs, so the Cypremort Reef right here, so there's a little bit of delay on this pointer, Cypremort Reef here at the mouth of the Atchafalaya, the Cali Reef, the Grand Isle Reef here on the right. These reefs are all in the vicinity of outflows of major rivers in coastal Louisiana. Obviously, the outflow of the Mississippi River here over on the right, particularly in the spring of this particular year, was really, really high. And so that certainly played a role in driving the seasonal effects that we have in the reef assemblages comparing fall and spring sampling event. And the same thing with the Atchafalaya, the Cypremort Reef at the mouth of the Atchafalaya. We actually got paddlefish DNA, and this reef's about 5 miles offshore, which is clearly derived from the Atchafalaya River and its outflow during the high spring flooding events. So it seems as though seasonality, particularly flooding events, is a big driver of the fish assemblages in these near or inshore coastal reef environments. What about material? So again, that was also significant. So in this case, we looked at paired comparison, which reef materials were different from one another. And really the only significant differences we got were when we compared the oil and gas reef to the concrete reef, or oil and gas reef to the limestone reef habitats. All the other reef materials were not different from one another statistically. And we look at the NMDS, these are the same positions of the reefs as the previous slide, but this time we highlighted the oil and gas reefs, sort of they cluster together in this NMDS plot. So it's just oil and gas that seem to be different. The interesting thing about that particular reef is it is the largest artificial reef in the world. I classify it as an oil and gas reef. It's really an old sulfur mine, and the support structures for that old sulfur mine off of the coast of this part of Louisiana called Grand Isle. But what I really think is driving that reef is the fact that it's in much deeper water than the other reefs. So it's really a different environment. It's in 50 to 20 meters depth, while all the other reefs are more or less than 5 meters in depth. So they're really different environments. Unfortunately, this is the only reef we have in the study that's an oil and gas reef. So to really say oil and gas reefs have higher or lower species richness, there really needs to be another study comparing those structures to one another in similar habitats. We just didn't have that option in this particular study. So if I come back to my original hypotheses, the introduction of reef structure will congregate fish from the surrounding area. The answer is yes, sort of, but reefs are not always more diverse than control habitats. The interesting thing is that Paxton et al. Published a study in 2020 where they showed the same pattern on a global perspective. This was a meta-analysis, not necessarily with eDNA, but with other sampling approaches, and they more or less show the same thing. We don't see big differences between reef fish assemblages and non-reef fish assemblages based on species richness values. And one of the things that resource managers really want to know is, are there more species, more individuals using artificial reef habitats in comparison to control habitats? And the answer is, we don't know, obviously. There's a big debate on whether you can quantify numbers of individuals based on a read number. I'm not a big supporter of it right now. I don't think we're there yet. But redoing this study with sort of some other independent visual surveys that quantify numbers of individuals might be able to give us that number. Reef material will affect the fish assemblage or fish community present. The answer is yes, but in this context, only for the oil and gas reef. And as I mentioned, I think the environment, the abiotic factors, the seasonal component are playing a role in why Grand Isle, at least, is different than the other ones. It's in much deeper water. So we kind of need to expand on oil and gas structures, particularly because we have this substantial rigs to reefs program in the Gulf of Mexico. We really need to investigate that more. And finally, will older reefs have more established communities? The answer is no, at least on our data, but this is based on we have a limited number of replicates. In our study, moderately aged reefs tend to have higher species richness. But again, I think expanding it to more reefs of different ages will really allow us to get at that question. Other things to consider, which we had to sort of deal with in the study, is turbidity of the environment. So there's on the right is one of the filters from our artificial reef filtering. In many cases, we couldn't even filter a full liter of water. We had to go through multiple filters to be able to get it all. So turbidity in our region is a big issue. The second thing, which I can't account for, I try to account for it as much as possible, is the effect of eDNA transport and drift. Our controls were 500 meters away, but unless you really model or test sort of the directionality of flow, this is a hard thing to wrestle with. I think we did a pretty good job of it, but I can't definitively say whether our controls were far enough away or not. And finally, I think anybody who works on eDNA sort of has run into this. The reference database for, at least for 12S and certainly other mitochondrial markers, is an underestimate. Everything is not sequenced, so we don't have a complete reference database, particularly for Gulf of Mexico fishes. So as part of the MyFish pipeline, you get a 97% or higher similarity matrix and an 80 to 96% data matrix. Again, we focused on the 97%. But what we saw is that on the bottom graph here, in the gray is the greater than 97% similarity matrix, and the black is the 80 to 96. When we see high or large numbers in the 97% data matrix, we also see high numbers in the 80 to 96% data matrix. And when I ran a correlation analysis, it's positively correlated. When one is high, the other is high. So you can see on average, for the control on the bottom left there, we got about 11 species on average across all the reefs of the control sites, and probably 12 or 13 at the reefs. When we look at the 80 to 96%, we're upwards of almost 30 and over 30 individuals. So our estimates of species richness at these reefs are truly underestimates of the true species richness at these sites, simply because of an incomplete data reference database. Okay, kind of switching gears. The second study focused on reefs in Lake Pontchartrain. And so, again, this was conducted by my student, Megan Reba. The title of this sort of subsection is Assessing the Impacts of the Bonnie Carey Spillway Opening on Lake Pontchartrain Fish Assemblages. So those of you who are not familiar, Lake Pontchartrain is one of the largest estuaries in the Gulf of Mexico. It's shallow, averages about 3 to 5 meters in depth. It's been subjected to a fair number of disturbances, urban and agricultural runoff. You see on the south side of the lake there, that's the city of New Orleans. Heavy overfishing in the 60s, 70s, and 80s, shoreline alteration, shell dredging, general loss of hard substrate in the lake. And one paper says that between 1900 and 1980, fisheries declined by 49% in the lake. So this sort of stimulated the deployment of artificial reefs in Lake Pontchartrain. And as of May of last year, there are 12 artificial reefs of varying materials, reef balls, concrete, and limestone, currently deployed in Lake Pontchartrain. We know a lot about the fish assemblages in Lake Pontchartrain based on this study from 2004. More than 97 species have been collected or occur in Lake Pontchartrain. So we have a good historical representation of the species that should be there, have been there. The only investigation of artificial reef diversity, fish diversity in Lake Pontchartrain was a report published by Mike Poirier from UNO. They conducted scuba, set gill nets around the reefs, and conducted hook and line fishing. And they collected 23 total species using those 3 approaches, but no single method detected more than 13 species. And obviously, using these approaches can be labor intensive and can be costly from a time perspective. More importantly, there's a high probability of missing rare species in these environments. You can see this is an underwater picture of Lake Pontchartrain. You can see how conducting a scuba survey here would be really challenging, and you would likely miss a lot of things. So this study was more or less conducted at the same time as the last study that I presented. We want to use eDNA to compare species richness across different reefs on the north shore and south shore of Lake Pontchartrain, comprised of different materials and different ages. That was the original goal. But then this happened. So there is an artificial connection between the Mississippi River and Lake Pontchartrain called the Bonnie Carey Spillway. So to the left, the downstream is where New Orleans is. If you follow the river up to the right, you go past Baton Rouge. The right side of the screen is where the water flows from Mississippi River through the spillway into Lake Pontchartrain when river water gets real high. The spillway was built to protect the city of New Orleans. There's a trigger of a discharge rate. When that's surpassed, the Army Corps of Engineers begins to open the spillway to release some water into Lake Pontchartrain to reduce the pressure on the levees downstream to protect the city of New Orleans. So we had done one sampling event and then got a bunch of rain, and then this happened. So the Great Flood of 1927 is sort of what stimulated the construction of the Bonnie Carey Spillway, as well as expanding the levee system in the central part of the US. This was obviously a major flood event. 500 people died, 700,000 people homeless. As part of the 1928 Flood Control Act, the Bonnie Carey Spillway, as well as other spillways along the Mississippi River, were developed as controlled spillways to minimize damage from flooding events during high water events. So here's the spillway here. Right here is the spillway. So it's a few miles upstream of the city of New Orleans, upstream of the Mississippi River. At the river, it's about 7,700 feet wide. Here at the lake, it's about 12,000 feet wide. And this was constructed in completed in 1931. Essentially, it consists of these wooden pins, 350 pins along the length of the spillway that are simply manually pulled by a crane when they open the spillway. When they open the spillway, they don't always open all of these pins or pull all of these pins. It's simply depending on what the flow is in the river. But if they have to pull all of them, it takes them 36 straight hours to pull all these pins to allow all the bays to be open. The trigger is 1.25 million cubic feet per second in the river when they start to open the spillway. So we know a fair amount what happens when the spillway is opened. Obviously, we get a ton of Mississippi River sediment flowing through the spillway, sort of settling. And typically, we find it most often in the southern portion of the lake based on the cycling of the currents in the lake. So we have an influx of sediment as well as an influx of fresh water during these opening spillway events. Of course, there's a lot of nutrients from the Midwest that are in the river. So we often have substantial blue-green algae blooms followed by fish kills when the spillway is open, particularly in the spring and summer months. But what we don't know about are the fishes. What happens to the fish community when the spillway is open? So again, this wasn't the original goal. The original goal was to look at the reefs, but this is an opportunity we can sort of assess, use eDNA to examine if the fish assemblages change. So we had 3 sampling events: before the opening of spillway, during the opening of spillway, and after the closure of the spillway is sort of when we did our sampling. Because we increased our sampling, we decreased the number of reefs, which we analyzed for this part of the study. So we had 3 reefs on the south shore: Lake Town, West End Pier, and Lakefront. These are limestone and crushed concrete reefs. And again, we chose these reefs because based on previous satellite images of the opening of the spillway, we knew that there could be substantial impacts there with increase of fresh water, increase of sediment in these environments. So that's why we focused on these 3. In terms of when we sampled, we had one sampling event that was 4 months prior to the opening of the spillway. Then when we found they were going to open the spillway, we went out there that morning as they were opening it. So we consider that a before open spillway. The black line shows the discharge rate through the spillway. This was the first time in history that the spillway was opened, closed, and reopened in the same year. So it was open from basically February through April, closed for a short while, and then reopen again from June all the way through September. So we had 2 sampling events there, and then we had 1 sampling event 2 months after the closure and 4 months after the closure. So it's sort of a nice series of sampling before, during, and after the opening of the spillway. We used the same approach as in the previous study: metabarcode DNA. Van Doren sampler, 3 water samples, 3 reefs, water samples, 3 water samples from each reef, 3 water samples from each control, abiotic data using the Van Doren sampler, same type of analyses as in the previous study. So what do we find? We collected a total of, or detected a total of 64 species across all of the sampling events. So there are 18 total sampling events across these 3 reefs and controls. Controls 18 to 38 species, reefs 19 to 43. The highest species richness was at Lake Town during the last sampling event 4 months after the closure of the opening of the spillway, or 4 months after the closure of the spillway. In 6 of the 18 sampling events, controls had higher species richness than did the reefs during our sampling. What about the assemblage? Did it change? How did it change? So here's the sort of we coded the species as freshwater or marine or brackish. So the black is marine, brackish, freshwater is the hatched. So Lake Pontchartrain is typically low salinity environment, 3 parts per thousand on average during a normal year. And so we basically detected what I would call a typical estuarine fish assemblage, anchovies, menhaden, speckled trout, the typical players you would expect to be there during our pre-BCS open sampling event. During, as you might expect, we started to receive eDNA from more freshwater species. We started to detect minnow or leuciscid DNA in our sampling during BCS opening event. And after it closed, up to 4 months after, we were collecting darter DNA. There's no darters that are known from the lake itself. They're in the river. More centrarchids, lipomus, micropterus DNA. They are present in the lake, but typically not in large numbers. So the proportion of freshwater species increased throughout the study period, simply with the influx of fresh water. In terms of looking at the assemblages, the shared species, so 28 species were shared across all sampling events. 13 were unique to the pre-BCS opening, none during, and 3 after. And you can see the 19, 0, and 1 represent species that were shared between different paired sampling events there. The greatest number of shared species besides all 3 was between the during and post-sampling event, with 19 species were shared. If we look at this in a multivariate context, so these are going to be divided up by sampling events. So 1 and 2 is before the opening, 3 and 4 is during, and 5 and 6 is after. So here is the pre-BCS opening, reef and control structures samples all grouped together. The day it opened was still before BCS opened, kind of the same thing, but different than the initial sampling event 2 months earlier. And then pretty much everything else, the during and after, all grouped together in an NMDS plot. And statistically, when we look at this, we see no difference between reef and control assemblages using in a PermaNova analysis, but we did see significant differences in sampling events. So when the samples were collected, we see different results associated with sampling event. Which sampling event? So we ran a pairwise PermaNova, and basically the only comparisons that were significant were 1 and 2 versus all of the other sampling events. In fact, 1 and 2 were statistically different from each other in addition to everything else as well. And so we look at this, sort of regroup this based on that analysis, we see 3 distinct fish assemblages associated with sampling event. What about abiotic influences? So we ran a best analysis using the bioenvironmental feature in primer, basically looked at the impact of individual abiotic factors on fish assemblage structure. So this NMDS plot is the same orientation of the plot before, except that the bubbles, the size of the bubbles, correspond to a particular abiotic feature. In this context, this graph shows salinity, because salinity was the only significant abiotic factor playing a role in assemblage structure. And when you look at the actual raw data, you can see here that we're between 1.13 salinity all the way up to almost 3.5 during sampling events 1 and 2. But as the spillway was open and we have this influx of fresh water, much, much lower salinity values. The highest we got was about 1.5 in lakefront, during the last sampling event. So it seems as though salinity, or the lack thereof, is driving the fish assemblage during the opening of the Bonnie Carey spillway. So today, this has been the most comprehensive summary of artificial reef fish diversity studies in Lake Pontchartrain. The previous study, we collected 23 species were known to occur at the reefs. We got 64. Now, obviously, this is somewhat artificial because now we have river species being detected at the reefs. Reefs generally have higher species richness than open water controls, but not necessarily. Again, if you build it, will they come? For the most part, they are higher, but not always. And statistically, there was no difference between open water controls and reef structures. So the opening of the spillway and the influx of fresh water restructured the fish assemblages. Even 4 months after the closure of the spillway, the community did not return to pre-opening assemblages. Salinity, as I mentioned, seems to be the driver in the fish assemblages in these habitats. I should say that we also have 4 sampling events for 3 North Shore reefs, so we can look at seasonality as well. I'm just not presenting it here. We were just interested in the opening of the spillway during this particular presentation. More interesting is the opening of the spillway is becoming a more frequent and regular thing. So in the first 60 years of its existence, it was only opened 8 times total. In the last 14 years, it's been opened 7 times, including in 2019 was the first time in history that it was opened and closed twice in the same year. So this is becoming a more frequent and regular event in Lake Pontchartrain and the opening of the spillway. So what are the goals of the reefs? This is sort of what managers need to think about. Is the goal to attract marine and estuarine fish assemblages for recreational fishing? If that's the case, then future reef construction events might want to focus on the eastern side of Lake Pontchartrain. The salinity over here tends to be a little bit more stable because it's under the influence of Lake Bourne and the Gulf of Mexico. It goes to the Wrigley's here. Salinity tends to be more stable in this environment. So if the goal is an estuarine fish assemblage, then these reefs need to be focused on that part of the lake. The other ones, they're going to be subjected to future BCS opening events and will be restructured clearly over time as the influx of fresh water occurs. Finally, just a couple things of what we're working on now. As I mentioned, the barcode databases are incomplete. So we have been trying to barcode as many coastal Gulf of Mexico fishes as we can using 12S mitochondria, the one that we've been using in the lab, the marker in the lab. But clearly, this needs to be done for other markers as well. Most recently, we started to compare fish assemblages on living shoreline reefs versus controls. We're doing this in Lake Calcasieu in western Louisiana in collaboration with Megan LaPere at LSU and Stephanie Archer at LumCon. They've constructed these oyster living shoreline reefs as structures to minimize wave energy, wave erosion, particularly in areas where there are important hurricane evacuation routes, like this area here on the right. But they want to know, are there differences in fish diversity associated with these oyster reefs in comparison to open water control habitats where there are no reefs? So we just started working on that this summer. And finally, one thing that I'm really interested in, I'm also curator of our vertebrate museum here, so I have a sort of a museum background. I've always been interested in trying to extract DNA from type specimens or valuable specimens, older specimens. And as you might expect, most curators don't want you cutting off a piece of a fin of the holotype or paratypes. So I thought, well, let me see if I can extract DNA from the ethanol storage fluid. This is only for ethanol fixed specimens, which are pretty much pre-1900 specimens. They were fixed and preserved in ethanol. They've never been in formaldehyde. So I applied eDNA techniques to the storage media, to the ethanol, to see if I could get DNA out of it and sequence it. And lo and behold, the answer is yes. I was able to get DNA from a specimen from 1860, one from 1838, and one from 1882. So 100 to 150-year-old specimens that was able to extract DNA and amplify it. This is using single species approach, using this technique. And to be forward and upfront, I did the same thing with the specimens collected by Charles Darwin on his voyage on the Beagle around the globe. I went to the British Museum of Natural History in London, and they let me filter the DNA from Darwin's specimens that he collected as fish specimens. So I haven't touched those yet because I want to maximize the protocol. Why am I telling you all this? Because one thing I want to do is I want to apply this approach to larval fish collections. Anybody who works with larval fish collections knows that when you get these large jars full of larval fishes, it takes a long time to identify them. So if I can extract DNA from a 100-year-old museum specimen, why can't I collect DNA and apply a metabarcoding approach from the storage medium of these larval fish collections to get more rapid sort of summary of the fish species that are present? So I want to apply a metabarcoding approach to this because I think a lot of people would be interested in sort of more of a rapid assessment of fish assemblages rather than waiting for months to years to get your larval fish identified. So I'd like to do a study. So if anybody's interested in collaborating on something like this, I'd be very, very interested. Finally, I have to acknowledge my funding sources: Louisiana Department of Wildlife and Fisheries for the first study, and Louisiana Sea Grant for the second bonded carrier spillway study, and a variety of people who assisted in various ways in both of these studies. And I'll stop there and take any questions.
Excellent. Thank you so much, Kyle. That was a fantastic presentation. I feel like we'd be really interested to bring you back and learn more about the ancient DNA work that you're doing. That is super cool. Okay, we do have some questions that have come in throughout the presentation. So folks, if other questions come up, feel free to type them into the questions for staff box. Our first one here is from Nicole. So this question came in the first part of your presentation. Was it difficult to sample the 15 to 20 meter depth by hand with the Van Dorn sampler? How did you deal with drift of the sampler at depth, potentially taking it away from the desired sampling point?
Yeah, so yeah, it's very difficult to do that. You have to apply enough weight that you can, I mean, that reef is really big. So even though if it drifts somewhat, it's easy sort of to get over that reef. But yeah, that's a challenge from the current perspective. We tried to put extra weight on the Van Dorn sampler, but of course that makes it more difficult to pull it up to the surface. But yeah, that wasn't as much of an issue because that reef is so big. We didn't have to get it exactly on the reef, just sort of in the vicinity, and we were still confident we got reef DNA from that.
Great, thank you. Okay, we have another question here from Hassan. He asks, how you define reeds? It seems you have doubt allocating the number of reeds to the number of individuals. Could you please elaborate on this? And I should have mentioned this question also came in the first part of your presentation.
I mean, I'm talking about sequence reads. Obviously, there's debate on whether you can use sequence reads as an abundance metric, since there's multiple copies of DNA of mitochondrial DNA per cell. How do you sort of correlate read number with abundance? That's when I talk to resource managers. That's one thing they always want to know, and I don't think that we are there yet in terms of using read number to assess abundance in sort of natural settings. It seems as though people are able to do it in sort of controlled aquarium settings, but in a natural environment, I don't think we're there yet.
Great, thank you. This one came in from Michelle in regards to the second part of your presentation. Is this BCS study published?
It is not. I have a manuscript that's really, really close. So my goal is to submit it within the next month. So no, it's not published yet, but we're pretty close.
Great, thank you. And I think folks could keep an eye on your website. I'm sure be updated with FuturePub. Okay, awesome. Great, we have a question that came in from Adam. So he asked, up here in New Jersey, multiple studies have suggested beach replenishment to be temporally, financially and ecologically disadvantageous. Hard structure and or combinatorial approaches may be more optimal alternatives. What's your opinion on limestone and or concrete-based strategies to supplement or replace beach replenishment, especially since these locations are typically in the shallower areas? He also followed up and said, wonderful presentation.
Well, thank you. Yeah, I mean, they clearly are going to attract fish. Those structures are, I would venture to, I mean, I don't know the exact cost of what some of those structures are. I don't do that, but my educated guess would say that those structures are economically feasible. It seems to be is like, what are your goals? What do you want in terms of attracting different fish communities, different fish assemblages in different environments? What do you want? Our data suggests that it doesn't matter what you put down there. So if that's the case, why wouldn't you put the most economically feasible structure there? Because fish are going to come regardless of what is there.
Excellent, thank you. All right, we have another question from Hassan. Could you use metagenomics instead of barcoding?
I don't know. I guess I really hadn't thought about that. I mean, obviously the barcoding seems to be the approach that everybody is using. I really hadn't thought about metagenomic approaches, so that's a difficult one to answer. I hadn't really thought about it, so I don't know.
All right, thanks, Kyle. Yep, sorry for the tough question here.
That's all right.
We have a couple other ones that are coming in. So this one is from Lou. What are your thoughts on using qPCR to enumerate fish populations, especially after identifying the species present at the reef?
Yeah, so some of the early eDNA assays that we did in my lab, we did use qPCR, and yeah, I mean, you could definitely use that. I think you'd have to have some pretty substantial experimental studies to sort of confirm those numbers are in agreement. Certain, quantity of DNA corresponds to, this. So I think from a biomass perspective, there's been a fair number of studies that suggest, yeah, you can use qPCR, you can use the eDNA approaches to look at biomass. But does that biomass mean one big fish or lots of little fish? That's sort of wrestling that apart is where the challenge is. So we did use qPCR for single species approaches, but now again we've moved into the barcoding approach to get more fish assemblage data, and that seems to be what most natural resource agencies are looking for. We use qPCR and standard PCR eDNA approaches when we were targeting some imperiled minnows and darters in freshwater environments a few years ago, but now we're sort of in the metabarcoding world. So yeah, I mean, if you really wanted to know a particular species abundance, I guess you could do it, but I would still be skeptical unless you really had a lot of experimental tests to sort of confirm the metrics that you were getting out of it.
Excellent, thanks, Kyle. Okay, a couple more questions here. So we do only have 2 minutes left, so I think we're only going to get to one of these questions. So I do want to mention that you can always reach out to Kyle directly. Kyle, I don't know if you want to just quickly flip back to your acknowledgement slide so they can grab your email.
Yeah.
Okay, and then this question, so we'll do this question from Becky. Extremely interesting presentation. You mentioned identifying control sites at 500 meters away from various reefs. Were there any other criteria that needed to be met to qualify as a good control, e.g. proximity to other environmental features, water chemistry parameters, etc.?
We simply wanted open water, sand/mud bottom environment, so limited or no hard, firm, benthic substrate. That was really the only other sort of criteria that we used. We wanted it to be, what you would expect in, like, for example, in Lake Pontchartrain prior to the construction of any artificial reef. Open water, we think of some oceanic and Lake Pontchartrain habitats as sort of an aquatic desert, open water, bottom, substrate of mud or sand. That's what we were looking for. So beyond that, there was no other criteria that we used to assess something that was a control or not.
All right, excellent. Thank you so much, Kyle. Folks, I'm so sorry. We do have other questions left, but we are at time at 1 p.m. Eastern time. So Kyle, real quick, just want to say thank you so much again for taking some time to present today. The Omics Seminar Series presentation was fantastic, and it clearly sparked some really good questions and discussion here. Well, and I also want to thank the whole audience for joining us today. Just a quick reminder that the NOAA Omics Seminar Series takes place on the third Wednesday of every month, 12 p.m. Eastern time. Our next seminar is actually in a few weeks on July 20th. That seminar is titled Global Marine Biodiversity Monitoring Through Partnership and Innovation, presented by Dr. Luke Thompson. More information on how to register on the NOAA Omics website. So see you all at that time. Thanks again for joining.